ladies and gentlemen. So again, we're going to be taking a look at. We're going to finish up our 3-7 number 2 notes. Nobody should have turned these in yet because again, like we're not done with them. So let me go ahead and get that opened for you, and we'll take it from there. All right. So in your notes, ladies and gentlemen, we finished up with function of RNA yesterday. This is so at this point, the only part of the notes that we should still be working on is the very, very last page where it says the 3 types of RNA. So this is where we are going to start off today. Let me go ahead and get the slideshow going. All right. All right, so the first thing I want to point out, ladies and gentlemen, is that last week we talked about replication. That is when DNA makes more DNA. DNA copies itself. This week, next week, and through the end of really before we hit the end of spring break, we are going to be talking about transcription and translation. Transcription is when we go from DNA to RNA. And then translation is the process of going from RNA to protein. If you recall, when I asked the question the other day on the assignment, what are the what are the 2 steps of gene expression? How do we go from DNA to an actual gene, or how do we go from a gene, I should say, to an actual protein that will express that trait? We said it was twofold. We said it was transcription and then translation. That's what we're going to focus on. Now, when we talk about transcription and translation, ladies and gentlemen, and again, this is from yesterday, we need to talk about the 3 different types of RNA. And for that purposes, ladies and gentlemen, we are going to go ahead and, boop. Oh, sorry, we already did this stuff. We have 3 types of RNA. So the first thing I want you to do, ladies and gentlemen, is it is in a. I don't know if the digital copy came out better than the paper copy that I did for my live students, but you do have this diagram somewhere in your notes. Again, I don't remember if it's on the right page or not, but there are 3 types of RNA. Okay, and you do have this diagram somewhere in your notes. So you have a blob looking object that is the ribosomal RNA, also referred to as rRNA. You will have a single strand RNA that is the messenger RNA. By the way, when we see pictures of RNA in textbooks, the messenger RNA is the classic traditional RNA, the single strand that's still a helix. Looks just like DNA, but it's only 1 strand. That is actually messenger RNA. And then we have this stuff called transfer or tRNA. And it kind of looks like a weird looking fork. Okay, so that's sort of what it looks like. So go ahead, take a minute and label your diagram: rRNA, mRNA, tRNA. By the way, very rarely do scientists actually spell out or write out or say ribosomal RNA, messenger RNA, transfer RNA. We use the shorthand. We say rRNA, mRNA, tRNA. All right, so these are our 3 types of RNA. So let's talk first about messenger RNA. This is numbers 1. I'm sorry, A. Underneath A on that last page, this is letter B, numbers 1 and 2. So your messenger RNA is exactly that. It is a messenger. So much like a messenger might pick up documents at a lawyer's office and carry them to the courthouse so that they get filed on time or whatever, the messenger doesn't really do anything with that information from the lawyer other than carry it. That's exactly what mRNA is about. It literally will just go from the nucleus to the cytoplasm with instructions. That's it. It carries the directions from the DNA to the ribosomes out into the cytoplasm. Because remember, the DNA will never actually leave the nucleus. So, since it doesn't leave the nucleus, but the ribosomes are outside of the nucleus, we need to have a messenger carry the message from the DNA to where those proteins are actually going to be built. Because remember, proteins are actually built in the ribosomes. All right, so again, part B on that last page. This is numbers 3, 4, and 5. So please go ahead and copy this down. Remember, when we take the message, this mRNA that gets created in the nucleus, it carries instructions in the form of codons. Remember, a codon is a series of three nucleotides that will code for a specific amino acid. And an amino acid, of course, are the building blocks of proteins. Okay, but the very, very first codon on an mRNA does not call up an amino acid. It is simply called a start codon. And we talked about these previously when we looked at that amino acid wheel. A start codon is like a signal to the ribosome, hey, start making my protein here. That is the point where the mRNA actually can attach to the ribosome. And it basically just tells the ribosome, hey, this is where we're going to start building this protein. Because keep in mind, the mRNA is not a full copy of all the DNA. It is literally just going to be the number of nucleotides that we need to build a specific protein. All right. I always tell people mRNA is like a single word in the dictionary. DNA is like the entire dictionary. If you just needed to know one definition, you wouldn't read the entire dictionary. You would just flip to the section where the word that you need the definition for is, and you'd look at it and read it and figure it out. That's exactly what mRNA is. mRNA is just one section of that DNA to make a specific protein. All right, so we have the start codon. After the start codon, we have a series of nucleotides, again organized in codons, that then tell the amino acids to come to the ribosome in a specific order. So we have a specific set of nucleotides that call up specific codons that will call up specific amino acids that are going to build in a certain order that are going to build up a specific protein. Now, that protein might be the protein that you need to digest milk. That might be the protein that you need to have a higher immune response. That might be the protein that you need to, have melatonin in your skin. I'm not sure what protein it will be, but it will only be for a specific protein. On that mRNA, the very, very last codon, again, does not call up an amino acid. It is the stop codon. And it is the signal to the ribosome to then stop production of the protein. It says, okay, you can stop now. It's kind of like the period on the end of the sentence. It's like, boop, this is where we end. Okay? Issues or questions? All right. You have a diagram of transfer RNA. Please make sure that you label it. It is towards the bottom of page 3. All right, so transfer RNA. Transfer RNA is our second type of RNA. So if messenger RNA is basically telling us it just carries a message from the DNA to the ribosome. Well, we have to get those amino acids onto that ribosome somehow. That is where transfer RNA comes in. Transfer RNA on one end has something that we call an anticodon, And at the opposite end, we have an amino acid. Okay. Now, the bases on the anticodon are complementary to the mRNA. So remember when we talked about DNA and then it has a second strand and it's complementary? They weren't identical. It was like peanut butter and jelly. You already kind of know which ones go together. So the anticodon is the complementary bases that match to the mRNA. The transfer RNA basically then reads the message of mRNA and goes and gets the specific amino acid that it needs at that time. So we talked previously about how DNA is like a recipe card. And then we talked about how mRNA is basically just like the information. So if you think of DNA as a recipe card and RNA is the chef, well, mRNA is when the chef reads the recipe card. tRNA is when the chef is running around the kitchen getting all the ingredients. Okay? So again, we can see in the diagram, we have an anticodon at the top. We have the actual transfer RNA in the middle, and then we have a binding site on the other end for the correct amino acid that we need to build the protein. This is 3, 4, and 5, still under transfer RNA. So transfer RNA then transfers the amino acids from the cytoplasmic pool to the ribosome. So, ladies and gentlemen, in every cell of your body, in the cytoplasm, there are some amino acids literally just floating around, waiting for transfer RNA to call them up. Okay? This is, again, if we're going back to that analogy of the recipe card DNA and the chef being the RNA, this is equivalent to a chef having a fully stocked kitchen, right? Like every time the chef has to make a recipe, he doesn't have to run out to the store. He just goes into the shelves or the pantry, or maybe he's got stuff on the counter or whatever, right? He's got all these ingredients laying around. He just has to collect them up. That is what transfer RNA does. Transfer RNA goes out into the cytoplasm and collects up all of these amino acids. By the way, these amino acids that are hanging out in your cell, you get these from the food that you eat. Or the dog or he, whatever. All living things do these. So any living thing is going to get these amino acids from the food that they eat. That they just hang out in the cells. Again, so each cell keeps a stock of all 20 amino acids. And then the tRNA attaches to one of the amino acids, and then it just will carry it over to the ribosome. So in this picture, the tRNA kind of looks like a little plunger, and then the brown picture, the big brown blob, is the ribosome. Issues or questions? All right, this is the last part of our notes, ladies and gentlemen. We are just going to be defining ribosomal RNA or rRNA. Again, this is very similar to the picture that we had earlier where we can see the transfer RNA, the ribosomal RNA, and then that strip, which is the messenger RNA. Now, I do want to point out at the top of this picture here, at the top of the first plunger coming in, that little blue marble looking thing, that is supposed to be an amino acid. And then over here, we see a chain of amino acids. That is where we're actually building up the protein. You can call this a protein, or sometimes we call these, because it's not actually folded yet. Proteins have to be folded. So right now it's not folded, so it's just a polypeptide chain. Anyways, ribosomal RNA is found, obviously, like the name suggests, in the ribosome. And what ribosomal RNA's job is to make the messenger RNA and the transfer RNA. It is a place where these guys are going to come together and it's going to help bind together the protein. So without the rRNA, like, the messenger has the message, the transfer RNA has the ingredients, but the ribosomal RNA, again, going back to that cooking analogy, the rRNA is like the actual preparation of the meal. Now I have my instructions, I have my ingredients, but now I have to put it all together. That is what the rRNA does. It puts it all together. It creates a place where the tRNA and the mRNA can come together, and then we can see that protein, or actually polypeptide chain, it'll eventually become a protein, is going to be put together, and then again, eventually, once we hit the stop codon, we stop transferring in new amino acids, and then it goes off to get folded. Any issues or questions?
No issues or questions.
Alrighty, ladies and gentlemen, I'm going to go ahead and stop my video.